Okay, so welcome back. We're on the second video of genetics, and remember, this is just overview. If you really want to get in depth in genetics, open up a book on genetics. Find a little like Cliff Notes book or something like that. The last section was just kind of a reintroduction to DNA, mitosis, meiosis, transcription, and translation when you're going to make proteins in the body. Remember, everything in your body is built from proteins, so it's important to understand how you make them. What are the two major processes in making a protein? You take DNA and you make a copy on a recipe card. What's that transition called? Where you write it down. It's called transcription. And then once you have it written on the card, you laminate it, you protect it, you move it to the cytoplasm or the rough endoplasmic reticulum, and you do what with it? When you take it from the original language and you convert it into this new language that you can use, you translate it. So translation. And then we make a protein. So remember, the central dogma is DNA to RNA to protein. It's the whole process. If you have any changes to that process, you have a dysfunction. If that protein was important for building your skeleton and your genes are missing for the recipe, what's going to happen to your skeleton? It's not going to be built right. It's going to be weak. It'll probably buckle. It'll fall apart. It's not going to work properly. Every bit of you is made on these proteins or dependent on these genes making proteins. Right? So now what we're going to do is we're going to learn some more principles of genetic change. Right? And then we'll talk about how diseases are altered to those changes. So it's kind of interesting because this is not just applying to humans, it's applying to lots of things. In the humans, we can see lots of genetic abnormalities, things like sickle cell anemia that you already looked up. You can look at something like albinism, when they're missing a gene to make pigments for color in their skin and their hair, and potentially even in their iris. Okay? Or things like polydactyly, where the genes for coding the number of toes or fingers were inappropriate, so they have an extra finger or extra toe. We'll talk about some of those disorders. But the same thing with fruit. I'm sure you've been in the grocery store and you see that one fruit that's really strange or looks abnormal. So something like a tomato with the extra little pieces bulging out, or a cucumber that's actually like Siamese cucumbers. So you've seen all these. These are all genetic flaws. So they grew out of each other basically because of a genetic flaw. And we're going to talk about how some of these flaws work. And we're going to stick, of course, to the human genetics. So you already figured this out, but how important is just one single codon? Extremely. When you're looking at sickle cell anemia, and I'm probably giving away the answer a little bit too much here, but when you look, the hemoglobin inside the red blood cell is about 120 some amino acids long. All it takes is one amino acid flaw, one codon to be flawed, and then the whole thing is defective. If the temperature is too hot, too cold, if there's too much oxygen, too little oxygen, if there's too much acid, too little acid, any of those changes make the hemoglobin molecule bend in weird shapes, causing the whole cell to bend strangely and cause them to stick together in sickle cell anemia. Right? And then other things we'll talk about, like cystic fibrosis. All it takes is one codon to be wrong, and they can't move chloride efficiently. So it can be things inside the cell that go wrong because of that one codon. It can be things on the surface of the cell that go wrong. It can even be the presence of the proper cells can be wrong, like in red-green color blindness, and we'll talk about this later too. Some people are just missing the appropriate pigment because they don't have appropriate genes to make pigments that detect red or detect green. And we'll talk about that. We'll talk about how you get that. So why is it that very few women get red-green color blindness, but lots of men get it? So few things are absolute. Mutations are the same thing. They're not always absolute. When you get a mutation to a cell, there are basically two major issues that can go on with mutations. You either change a somatic cell or a germ cell. If it's a somatic cell, when you change it, it's changing you, the person. When it's a germ cell that mutates, it's changing your offspring. It's affecting your offspring. So when you go in to get x-rays, if they zap your reproductive structures, they can actually damage those reproductive cells and cause mutations that would affect your offspring. So mutations don't cause you to shoot laser beams out of your eyes or grow bone claws or anything like that. That's not a typical mutation. Mutations also aren't always fatal. They're not always dangerous. Mutations are what cause evolution. So you have to keep in mind that mutations can potentially be good. All right? So some of these things, like mutations, are happening all the time. Every time you replicate a cell, for every 1,000 of those little letters, the base pairs, the BP stands for base pairs, for every 1,000 letters that you have to copy, you have one mutation. You have one flaw in that. So when you have 3 billion of those letters, you're getting lots of mutations every time you make a brand new cell. Accidents are bound to happen. We're going to talk about cancer next week and what happens when you start accumulating these changes. Sometimes the mutation can happen to your germ cell or your gamete after it's left your body. So let's say that as the gamete's leaving your body, a sperm's leaving the body, or even an egg moving down, If a mutation happens to something like eye color, you may have a child that has a strange eye color. Like if you've ever seen people that have two different eye colors, one's brown and one's blue, a mutation happens somewhere in here that affected their genes. Right? So germ cells versus somatic cells, the big picture. Right? So typical causes of mutations, there are three major causes. Spontaneous, which means it just happens. Remember, every time a cell replicates, it has about one mutation for every 1,000 letters it copies. So those GCs and ATs. Radiation can do it. So things like getting zapped by an X-ray can change it. That's why we don't X-ray pregnant women. And that's why we don't get lots and lots of X-rays. We're pretty limited to the number we get. And then also things like UV radiation can change the cell structures. And then chemically induced, whether it's chemicals in the environment, that person smoking right outside the door of your favorite restaurant, the person actually inhaling this. So chemically induced. And we'll talk about some of those, especially next week when we talk about cancer. So spontaneous changes can be when normal replication just goes wrong. Radiation induced, exposure to things like ultraviolet light, X-ray, and then chemicals in your environment. So what are the potential outcomes? So usually the outcomes when it's affecting your DNA is that you have some kind of a loss. Usually. So you can get gains which cause things like tumors or abnormal growths, but a lot of times what you'll see is when you cause a change to the DNA, you lose the ability to make something. So you may lose the ability to repair your DNA. You may lose the ability to control that cell cycle, which we'll talk about next week. You may lose the ability to transcribe things. Some of those changes may actually lead to apoptosis. And if you remember that term, what is it? It's cell death, cell suicide. Right? Some other outcomes, tumors. So here you can see that smoker, nice pink lungs to the smoker's lungs. You can see kind of cancers, which I should put a little lump in here, but here's skin cancer that's happening on this woman's nose. I'm assuming it's a woman. And then potential death and disability. So those are some of the possible outcomes. And I put this little kind of review or overview slide. There are a lot of changes other than those basic three. Cell metabolism can cause a spontaneous change. So if the cell's not making energy properly or making free radicals, that can cause it. Viruses can get in and change the DNA. Radiation, chemical exposure, replication areas. So those are all good examples. Right? Some of the outcomes, you can see. Cell cycle doesn't work properly. They can't transcribe properly. They can't repair DNA properly, and then eventually they want to die. Right? So DNA changes that lead to mutations. Here are kind of the major types of mutations. So we saw some of the causes and we saw some of the effects, but now looking at the DNA, the major changes. So first you can have a base pair substitution, or a frame shift mutation, or an overall chromosome change. We're going to focus on these two right now. We're going to talk about chromosome changes a little bit later. So a base pair substitution, I'm going to show you on the next slide, right, is when you take that whole long alphabet. So here's your DNA. You can see all of the letters, and we just cut out a section of these letters right here. And you can see that code, and this is in your textbook. If you take one letter and you substitute with something else, you change what this whole word is. Remember, this codon is now changed. So if you had like this sentence up here, let's say it translates as the big fat dog and the red cat ran for one day. In a way, it kind of makes sense. It's a smooth sentence. It may not be the most interesting, but it's a smooth sentence. If you change just one letter in one word, it can actually change the meaning of the sentence. So, or the way the sentence reads. The big fat dog and the red cat run for one day. If you're lucky, you can still get the idea. And this is kind of cool because you can see this one amino acid has 3 different words that tell your genes that amino acid fits. So if you're lucky, maybe you replace one of these letters and it still comes out with the same amino acid. That protein will still be exactly the same. So when you looked at something like sickle cell anemia, that one amino acid that changed the whole protein, if that one amino acid is still the same, even though the 3 little letters that wrote for it, like this, change, you're still in good shape. You still have a normal cell. It's when you change it to the point where you don't understand, like ren, what does that mean? Or ren. You don't know what those mean. So it can change the meaning of the sentence. In this situation, each of these words, if one changes significantly, it can change the shape of the protein. And the protein doesn't work anymore. So you can substitute, replace, exchange, and that's one problem that could happen. This is potentially dangerous, but it's not as high of a risk because remember, you're only substituting one of 4 letters. So if you have this one original letter, there are only 3 other possible options. So you might get lucky and it still is readable, like this. Right? The next one's called a frame shift mutation. These are the more dangerous because a frame shift, what happens is you lose an entire letter. So when you lose the letter, it changes how the whole sentence makes sense. Remember, every word has to be 3 letters long. So the big fat dog and the red cat ran for one day, if you lose just one letter, it changes every word beyond that. The big, and then look, 3-letter, 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 all the way along. So it's like unlegible. Same thing as if you put a letter in that doesn't belong there. That's still a frame shift mutation. So let's say I add an extra A. I stuttered a little bit. The big fat, right? It still changes everything beyond that. These are dangerous mutations because all it takes is one letter to be added. It doesn't matter which of the 4 letters it is. As soon as you add one or subtract one of these letters, it changes the entire protein, everything beyond that. Every one of these amino acids is changed. So the more dangerous is the frame shift mutation because it will cause dysfunction. Right? And then we'll talk about this more later, but chromosomal aberrations. These are changes to a whole chromosome. So you can take that one gene and instead of changing just one individual letter, you can actually take the whole gene and you can remove it completely, which means they don't have the recipe. It's like going to grandma's house and when she's not looking, you rip the whole page out. Well, what if grandma wants to make that recipe later? It's gone. She can never make it again. What if that protein in your body, that gene, was important for repairing your cells? You can never repair that cell again because you lost the gene. Another possibility is when you relocate. You take it from one chromosome and you move it around to another one. Well, now it's like taking one page out of one of your books and just randomly shoving it into another book in your collection. It's not going to be easy to find. You're going to have some problems because of it. Sometimes you can get a gene that duplicates, which means that gene kind of stutters. So when you make one, you don't just make one copy of this recipe, you make 2. So in other words, every time you want to make one batch of cookies, you end up making twice the batch of cookies. So you get a lot of cookies that way. You want to make one protein in your body, poof, you make more than you need. You make twice as much as you need. So those are some entire chromosome problems that we'll talk about later. All right, so what? Oops, that doesn't belong there. Sorry. There's the homework question. So homework question number 4. What are the 3 major DNA changes that lead to mutation? And I just went over them. Okay? And which one are the ones that we call a point mutation? So in other words, which one that changes just one letter, that's a point mutation, is typically worse and unforgiving. And why is it so unforgiving? So if you need to stop, rewind, and re-listen to that little section, go back to it because that's a super important point we're going to keep talking about. All right, so examples of mutations causing disease at the molecular level. The biggest key you want to remember is that when you change a protein, you either change a structure or an enzyme in general. So when you change these proteins, you may make a non-functioning enzyme which blocks a metabolic pathway. You might make a structure that makes too much or a bigger size cell. Or you might not be able to make a structure that causes the cell to be weak and fragile. Right? So these changes can be anything from inconvenient to lethal. And I'll give you some examples here to show you. So how to mutate a cell in just 4 easy steps. Sounds like a book to me. But a mutated gene codes for some kind of non-functioning protein or an enzyme. So remember, it had a purpose before. You take your DNA, make an RNA, there's your RNA, and you make some kind of an enzyme. Enzymes speed up reactions. So if we just use some random kind of chemical, we'll just say lactose. So you have an enzyme that breaks apart lactose. And if that enzyme is not there, can you break apart lactose? Nope. So lactose is a sugar. It gets broken down into 2 monosaccharides. If you don't have that enzyme, you can't do it. So people that have a genetic problem where they can't make this RNA, they can't make the enzyme, they're called lactose intolerant. Right? So they can't go through that reaction anymore. Then what happens is that you accumulate that chemical, like lactose, in your GI tract, and it makes you sick or nauseous. Lactose intolerance I've never heard of causing death, but yeah, it usually causes some kind of a sickness. Right? So let's give another example. This is kind of more of an inconvenient, not usually lethal, but a model enzyme for pathophysiology would be something called the fast flush response. And this is kind of interesting because we talked about this pathway a little bit before, where you drink alcohol, alcohol meets up with an enzyme that turns into acetaldehyde. Okay? Acetaldehyde is actually what gives you that drunk feeling, that intoxicated feeling. Alcohol is more of a toxin, it's actually really dangerous. The acetaldehyde makes you feel really uncomfortable, that like drowsiness, this sleepiness, the stupid feeling you get when you drink alcohol, right? And then that acetaldehyde gets turned into aldehyde dehydrogenase. And that basically turns into citric acid or acetic acid, right? So that acetic acid's pretty much just vinegar, and then you urinate it out. That's the normal process. You drink tequila, you feel a little bit of that feeling, and then you urinate that stuff out. Well, if you're missing this enzyme that converts acetaldehyde to acetate or acetic acid, you can't make it into the vinegar and urinate it out. So the acetaldehyde builds up in your body. The alcohol, the ethanol build up in your body, and you feel really sick really fast. This is called the fast flush response. It happens to more of the Asian population. About 50% of Asians are missing or have deficiency in this enzyme. So when they drink, they get like really drunk and kind of sick really fast. Their blood pressure changes because it's a vasoaffector. So it makes them feel a little bit more drunk really quick. It's called the fast flush response. And because it vasodilates, it makes them look a little bit more flush. Right? So it's just an interesting pathway. Right? Here's where it becomes lethal. Tay-Sachs affects basically catabolic enzymes. So catabolic means to break down. So it affects enzymes that break apart lipids. Here you have a nerve cell. Inside the nerve cell you have these little lysozymes, which remember are like stomachs of the cell. The lysozymes will break apart lipids, process the lipids, and actually help you make lipids to put in your cell membrane. Well, the problem is in Tay-Sachs they're missing an enzyme to break apart the lipids. So they accumulate lipids inside this neuron. As they accumulate, they start bulging out the lysosome. They start filling it up. The lysosome is going to start making these little pockets of lipid all over the cell. Well, here's the problem. Lipid's an insulator. So what's lipid going to do to a neuron? It's going to insulate the signal and make it so that it can't carry the signal appropriately. Insulating a signal on the outside of an axon is good because it speeds it up, but if you're blocking on the inside, it's bad news. So these children, they're born with this genetic disorder. It's called Tay-Sachs. And what happens is they start developing normally for the first couple of years, so they're learning to walk and talk, they're feeding themselves, and about age 1.5 or 2 years of age, these neurons in their brain aren't working so well anymore. So they start slowing down, they start regressing, they start forgetting how to walk, forgetting how to talk, forgetting how to feed themselves. And usually by the time they're 5, they've died. So Tay-Sachs is a really dangerous disease. And one of the reasons I'm bringing up Tay-Sachs now is because a lot of these genetic disorders actually run in families. So if you have a tendency to have this disorder, it might be because your family has it. There's a group called the Ashkenazi Jews that traditionally they prefer marrying other Jewish people that are just like them. It's a religious belief. So the problem is that when you have a disorder that runs in this community and you keep remarrying people in the community, the disease stays in the community. So they found that actually about 1 in 27 German Jewish population are carriers for Tay-Sachs disease. I remember watching a video a long time ago when I went to Iowa State about this, and it was just like tear-jerking. This family, they didn't realize they carried the gene, and they had a child that had Tay-Sachs, and it passed away by the time it was 5. And then they had another child, and it had this exact same thing. And then when they were tested, they realized they were both carriers. So we'll talk about what it means to be a carrier and how that affects your children. Yep. So in the general population, about 1 in 250 people are actually carriers for Tay-Sachs. So it's a really dangerous disorder, and it affects your ability to make enzymes that break down lipids. Right. Another situation, breast and ovarian cancer. They affect repair enzymes. So enzymes are important for lots of things. So this BRCA, it doesn't cause all breast and ovarian cancers, but it has a strong link. We know where it is. We know it's a defect in this gene, and if you don't have this gene, you can't make the BRCA. So this enzyme is a repair. If you have defects or mutations in your DNA, like it breaks, this BRCA comes along and it helps glue it back together or fix it. If you don't have functional repair mechanisms, you can't repair the cell, and the cell will become a tumor or a cancer. Right? So I don't want to point fingers straight at the BRCA because there are actually about 200 different changes that can happen that mutate this cell. So nothing's absolute again. Right? And it's not just BRCA that causes breast cancer. Right? Another mutation might be a structure. Not an enzyme like the last couple examples, but an actual physical structure. So a disorder called Marfan syndrome is interesting because it affects one structure, and it affects collagen that helps build you. So people with Marfan syndrome, what's interesting is that they make collagen that's not really strong. It's kind of defective, and they make lots of it. So when you look at them, they're really tall. Okay? They kind of look lanky because it's defective, right? When you look at their fingers, they call it arachnodactyly because they look like they have long spidery fingers. They usually get cataracts or malfunctions where their cornea will actually peel off, right? And then they have a lot of heart problems. We'll talk about this in just a little bit again. There was actually a president they believe may have had Marfan syndrome. He was really tall, very lanky, very awkward looking, long fingers, and if he hadn't been assassinated, he may likely have died of a heart attack early. And that was Abe Lincoln. So this is going to be talked about again when we talk about single gene disorders. So where do mutations usually occur? So this is our last slide, and I'll give you the next homework question. mutations can either occur by spontaneous generation, they just happen. So sometimes you just accidentally have a break. And a lot of times these spontaneous mutations are caused by things called mutagens. And mutagen means mutation generator. Things like UV radiation, exposure to chemicals, like toxins in cigarette smoke or toxins that are put in the water, right? So spontaneous mutations just happen. There's no genetic link that it was because of your parents or anything like that. And then there are other things called mutational hotspots, which means that some chromosomes are actually just weak in those areas. So the area of the chromosome has a high mutation rate. It changes a lot, right? So those are two places that you usually see mutations occur. And then the last question here for this section, homework question number 5. So a mutagen is a chemical or physical agent that causes genetic material to change. Oops, forgot a word there. Change. It mutates the genetic material. So in just a few sentences, remember, don't write me a book. What are some common known mutagens that are in your food? So what are some things that you eat that can cause mutations? Right? So when you find out that chemical, just tell me what mutation does it have a reputation for causing? All right, and just as an example, you can't use this because it's not in your food, and don't use this as an example, but in cigarette smoke, there are mutagens inside the cigarette smoke that actually cause things like lung cancer, right? GI cancers. So that's the first part of the question. The second part, what are some known mutagens in your environment? Don't use tobacco smoke. It's out of the question here. So then what does that mutation usually cause? Same idea. Something that's in the environment that you may have heard people freaking out about that may cause cancers. What cancers does it usually cause? Maybe you've heard some ads for lawyers, ambulance chasing lawyers that are saying, does your family have this disease? Well, it's caused by this kind of mutation. Right? There's an idea. And then the last one, what's another known infective agent? Mutagen. So what's something that can infect you? This isn't just an environmental toxin. It's not something in your food necessarily. It's actually some kind of organism that can get in you and cause mutations. And what do they usually cause?